coming to us from Columbia University. He got his bachelor's at Dartmouth before he went on to a PhD at Cornell in the Department of Neurobiology and Behavior. And then he went on and did a Miller Fellowship for his postdoctoral research at UC Berkeley, where coincidentally, when he left to take his faculty position at Columbia, I moved into the desk that he vacated when I arrived at Berkeley, which is sort of a fun little connection. I first became aware of Dustin's work when I was finishing my PhD, and he was publishing some really exciting papers on the role of environmental variation in the evolution of social behavior, which had some pretty interesting applications to the patterns that I was finding while studying social lizards. He's done a lot of comparative work across a very broad set of taxonomic groups, but as this year's UMMB Robert W. Storer Fund speaker, I'm hoping he's going to talk a little bit about his amazing work on African starlings, which I think are a great system for asking questions about response to changing environments. So without further ado, Dustin.
Thank you, Allison, for the invitation. It's great to be here. I realized I was here 10 years ago, almost to the week, I think, for the Young Investigator Symposium in 2007. And it's nice to be back. I have a soft spot for Michigan. My parents met here. My dad got his training under Dick Alexander. And then I went to Cornell when pretty much the entire department, both the neurobiology and the behavior people, all went through Michigan at some point. And I was reminded of that when I was back giving a talk last month. And I didn't even realize all the faculty on the neuro side, how many of them went to Michigan too. So there's certainly a whole generation of behavioral ecologists that have come through here, and it's nice to give you a chance to hear some of my work. So I'm going to talk a little bit about two things today, a little bit about behavior, but then a little bit more broadly about how animals adapt mechanistically to environmental change. When we think about global change and what humans are doing to the environment, we often think about the pollutants we put in the air and how this is changing temperature. So the famous Mauna Loa curve, looking at parts per million in the environment over the last 50 years and the effect that that's having on mean temperature. But one of the major consequences of global warming is not just changes in mean climatic factors, but also changes in the variance. So extreme weather events, storms, tornadoes, floods, are occurring more frequently now than ever before. We're seeing weather events in places of the world where we rarely do. Snowstorm from a few years ago in Israel, where I come from, we're experiencing massive 50-year storms more frequently than 50 years. Okay, and these are having a toll on our society and our infrastructure. But all organisms are having to deal with these extreme weather events and this increasing variability in climate. And I would argue that to understand how organisms are going to respond to these increasingly variable environments, we first have to understand how they cope with the natural levels of environmental variation. Okay, so there's many species that have evolved in parts of the world where they experience extreme climatic variation as a normal part of their daily lives, and they've experienced that over their evolutionary history. So what I want to do today is talk about some of those species. First, I'm going to use behavior as an example. I'm going to focus on social behavior from some African starling work to show how social behavior itself may be a way that animals have adapted to environmental change. And then I'm going to switch gears a little bit, continue to talk mainly about work in starlings, but explore some of the physiological, the epigenetic, and the genetic responses that organisms use to cope with these naturally variable environments that I mentioned. So we'll talk about sort of social behavior, and this is one of the starlings that I'll talk about throughout. So the group of starlings sternidae, there's about 120 species in the world. About 45, 50 of them occur in Africa. I'm only going to talk about the African ones today, and you can see that they're a very diverse group, both in terms of their ornaments. They've got this very flashy iridescent plumage. People in my lab study this a lot. They're one of the only group of birds that uses all of the possible ways to create these iridescent plumages that birds do. So they're kind of interesting from that group. It influences their patterns of speciation and diversification. But what I'm most interested in is their social behavior. And so about 40% of these species are cooperative breeders, which means that more than two individuals are taking care of the young. And 60% are your typical pair bonding species. They'll raise a clutch of young, and they'll disperse and breed on their own. So they're one of the more socially diverse groups of birds in terms of the number of species in the group that are cooperative. Now, one of the goals of my PhD work long ago, which I probably presented last time I was here, was to relate the incidence of that social behavior to the environments that the birds live in. Okay, so we published about a decade ago now a study showing that the cooperative breeding species tend to occur primarily in the savanna environments, and the non-cooperative, the non-social species tend to occur primarily in the forested environments. Okay? And what is it about those two environments? Well, if you've ever been to sub-Saharan Africa and you've been to the savanna environment, one of the striking features that you'll notice is that it's the climate plays an important role in shaping all that goes on there. Okay, this is a picture from the Serengeti, and the classic wildebeest migrations are driven by these cycling patterns of rainfall. Okay, and they influence so much of the lifestyle of all of the animals there. But one of the key features is that when the rains come, when the rainy season begins, is very variable from year to year, and how long they last is also very variable. So a bird has no ability, or any organism has no ability to predict when the rains are going to come and how long they last. And it's that degree of temporal variability, year-to-year variation, that it actually seems to correlate with the social behavior, okay? So this slide takes a bunch of GIS analyses together, but basically the cooperative species tend to be found in the more temporally variable, the less temporally stable environments than the non-cooperative species. And this was based on, again, this study of 45 birds, and for many years people would cite this as sort of the exception to the rule being that there are no good environmental predictors of social behavior in birds. Okay? We know that the environment influences cooperatively breeding behavior. We've known that for 30 or 40 years, but we tend to think about it in terms of resource distribution on the landscape, influencing delayed dispersal. But at these big sort of macro geographic scales, this study was the sort of the exception. So a bit later, we asked, well, what if you looked on broader taxonomic and geographic scales? And we looked across all birds. We excluded marine species, so we looked at all the terrestrial species that are likely to be, their food resources are likely to be influenced by terrestrial climate weather patterns. And what we showed was that if you look at the distribution of the cooperative breeding species, the red means there's more species here, they tend to occur in certain parts of the world, sub-Saharan Africa, Australia, New Zealand, and they don't occur in other parts of the world, particularly in the neotropics and the Indo-Malay Peninsula. It turns out that if you look at these distributional patterns and you look at the mean and the variation within and among years and try to see what predicts those distributions, the strongest predictor is the precipitation variation, and it's precipitation variation among years. Okay, so it's this temporal or year-to-year variability in precipitation that correlates most strongly with the distribution of these species. And when I say most strongly, these models predict about 14% of the variance, which is what you'd see in other types of life history traits that are predicted by these types of things. Okay, so again, on just starlings and across all birds, we tend to find cooperative breeding species in these drier, unpredictable environments. Okay, and more recently, we followed this up. So that study was done sort of before we had a phylogeny of birds, and here's the similar study, one of the results from that I'll talk about the study a little bit later, but if you look at environmental, in this case, harshness, so it's taking a principal component that takes into account temperature variation in means, precipitation variation in means, you find that the cooperative species tend to be in the more harsh environments than the non-cooperative species, and this is done of about 4,500 species and done in a phylogenetic context. Okay, so it illustrates again that in birds we tend to see these biogeographic patterns. And it's not just birds. A recent study from last month or so shows that we see the same thing in mammals. Okay, so here's the graph, here's the map that I showed you from birds, showing you where these cooperative species tend to occur. Here's a similar graph from mammals, and if you look in some of these places, particularly in sub-Saharan Africa, what you see overall is that the cooperative species tend to be found in areas with more rainfall variation and lower predictability in rainfall variation. So the same general patterns that we're seeing in birds, we see in mammals. Okay? But comparative analyses, I think, do a good job at giving you sort of broad patterns and generating hypotheses to test. In this case, we know then there's an association between climate and social behavior, but we can start to ask questions like, why do cooperative species typically occur in these harsher environments? What is it about living in those environments where there is some benefit to having helpers at the nest and perhaps living with family? There's really two non-mutually exclusive hypotheses. The first is that cooperation allows species to breed when non-cooperative species cannot, and cooperation increases the fitness of individuals in those groups, particularly when conditions are poor. And I'll show you a little bit of data illustrating each of these points. So one way that we can approach this is to do comparisons of different species. And so the species that I study the most is this superb starling. It's a cooperative breeder. I'll tell you about its lifestyle a little bit more later. But I also study this non-cooperative breeder. And this Hildebrand starling is a facultative cooperative breeder. It's mostly a non-cooperative breeder. Pairs breed, produce offspring, and they go off and disperse. But every few years, a small proportion of the nests will have a single helper. Okay, so we've got one highly social species, a non-social species, and basically a non-social species that sometimes can be facultatively social. And if you look at some of the reproductive life history traits among these species, they're consistent with this idea that I mentioned. Okay, so the cooperative species breeds for longer than the two less social species. Okay, its breeding season is nearly twice as long. It starts earlier in the year, and it goes for many weeks beyond that. And that's because it has many more multi-broods. Most of the non-cooperative species will only lay one clutch of eggs a year, but by having helpers, they're able to lay 2 and sometimes 3. The record is 9 clutches in a season, and that's because the predation is really high. So they get eaten, but they just keep laying. Whereas if these two species tend to get eaten, they very rarely lay again. And what's nice about this part of the world is there's actually 2 rainy seasons, and I'll explain why a little bit later. But the cooperative species is able to breed during both of those rainy seasons, and the non-cooperative species generally is not. The 2 years when they did, and when they had some things over here, was during an El Nino event, and the 2 rainy seasons sort of bled together as one super long rainy season. Basically, it never stopped raining, and they just bred in both of those periods. Okay? So as our breeding life histories are consistent with these potential benefits of being cooperative, we can also look at some of the costs. Okay, so here I'm showing you the oxidative costs, looking at reactive oxygen metabolites, comparing in the non-cooperative species birds during the incubation period and the chick rearing period. So as you move from incubation to chick rearing, these oxidative costs increase in both males and females. When you look in the cooperative species, you don't see this. Okay? When you look at males and female breeders in dark here and helpers in white, there's no change in their oxidative stress, okay, from incubation to chick rearing. And presumably, that may be because there are more individuals helping to rear the young and they don't experience some of those same costs. Okay, so cooperation enables species to sustain breeding during good years. They breed for longer, and they produce more broods per pair. They're able to reproduce in those harsh periods, and the superb starlings is during that dry period when the non-cooperative species cannot. And it seems to lead to a reduction in the cost of reproduction, in this case an oxidative cost of reproduction. Okay, so there are a number of lines of evidence by comparing these two species that are suggesting what some of the benefits may be to those that breed cooperatively in these environments. But I think what we really need to get at this question is understand how cooperation influences fitness. Does it lead to an increase in fitness? Okay, and so this is where I'm going to take you to the superb starling. The superb starling is a plural cooperative breeder. It lives in very large groups. Mean group size is about somewhere between 20 to 30 usually. There are usually 3 to 4 breeding pairs in the group. Each pair will build their own nest in a different tree, and then a large number of helpers, anywhere from 1 to 14, will help take care of the offspring. And they're essentially an obligate cooperative breeder, which means every nest has at least one helper. Both sexes help. Males are the primary helper, and females are the primary disperser. So we have a male philopatric society, with the kin structure suggests that the males tend to be more related than the females, although as we expand our data set, even that's not so simple, and this actually has very complicated kin structure dynamics that change annually for a variety of reasons that I won't get into today. But generally you can think of them like a classic plural cooperative breeder with male philopatry, and having helpers is sort of essential. And why is it essential? Well, they live in this environment. It's very seasonal. As you transition from the dry season to the wet season, this picture was only taken about 2 or 3 weeks after the start of the rains. As soon as it rains, it turns green, literally overnight. I was there teaching a class in February. They've been in a massive drought because of the La Nina, and we happened to get one night of heavy rain, and the next day you could actually see the grass green, and the students were amazed. So this happens literally overnight. But this transition, as I said, when the rains come and how long they last for, is completely variable from year to year. And so we describe this environment as being temporally variable and unpredictable. And the reason we have these rainfall patterns is because of the monsoon rains. And so this is just an animation of mean monthly rainfall. And you can see what happens is the monsoons move from southern Africa up over the equator and back down. And we're smack dab on the equator about here. And so we get a double rainy season and it moves up and down. And you also see this little cloudy area here. This is actually what's called lake effect rain. Okay, so we get a primary rainy season, which starts about now. It started this week, actually, in Kenya. And then we get a secondary rainy season here. And the length of the long-range breeding season can vary from 3 to 5 or 6 months. Whether they continue to breed in July and August depends on whether they get a pulse of rain in June. You get this second little peak here, and this is this sort of lake effect from the Great Rift Valley lakes. Okay? So we've got this double rainy season, but what it means is we also get 2 dry seasons, a very intense dry season, January, February into March, and a slightly less intense dry season a little bit later on. And when I mean intense, it often never rains in February and it often never rains in March. So it's very dry. If you plot the mean monthly rainfall per each month versus the variation in mean monthly rainfall, you would expect a relationship like this. But the point is the months that are the most variable are also the driest. And it's the 3 months before the breeding period, which we call the pre-breeding period. And this period is critical for most of the life history decisions that the birds make. Okay, so I'm showing you here how it affects behavior. This is the amount of rainfall that falls during a pre-breeding rain period over 4 different years, and it's correlated with the number of first-time breeders or the proportion of first-time breeders in the group. So in dry years, following dry years, you get very few subordinates trying to breed for the first time, and following wet years, you get many more. It also influences the amount of help that a helper does. They tend to help more following dry years and less following wet years. It affects numerous things of their life history. This is sort of an interesting one. This is, again, pre-breeding rainfall versus the proportion of male offspring laid in the immediately in the breeding season following. Okay? So after very dry years, females produce male offspring about 60-40, and in very wet years, they produce female offspring 70-30. And I just popped on some more recent data too, and you can see that this is a very robust trend. And it also influences their physiology. I'll tell you a little bit more later, but here I'll illustrate it. Influences their immune function. So again, pre-breeding rainfall, and this is a bacterial killing assay, so the ability of their blood to kill an E. coli bacteria in a dish. And that ability is heavily compromised in those dry years, and then in wetter years, it's not. And we see the same pattern when you look at breeders, helpers, and these non-breeder, non-helpers are birds that are neither breeders nor helpers. They're sort of intermediate dominance rank, and they don't play a reproductive role, although they do play a role in group defense. Okay, but the one thing that it doesn't, I mean, basically whatever you throw at that pre-breeding measure, everything sort of correlates with it except the critical value, and that's fitness. So reproductive success, no matter how you measure it or how you bin rainfall, are unrelated. So here this is breeding rainfall. I could have shown you pre-breeding rainfall, and I'm showing you mean reproductive success within a group, so averaged over these years. And again, you can do this in many different ways. And the fitness, the number of offspring produced and or survived to fledge is completely unrelated to the environmental conditions. And that puzzled us for a very long time. Okay? And it puzzled us because when we think about all of the theory in cooperative breeding behavior, we think about the role of mean fitness. Okay? So the classic sort of ecological or environmental hypothesis is the ecological constraints hypothesis. This idea that there's some constraint in the environment that limits a bird's ability to disperse, and rather than dispersing into a low-quality territory or where it can't find a mate or can't find a nest site, it should stay home and help rear its siblings. And this whole hypothesis is predicated on the idea that there are different mean fitnesses, W here, for different strategies: dispersal, floating, independent breeding, etc. Okay? Now, this really has dominated our thinking for a long time, but when the idea came out, in the late 80s, there was another idea that came out not long after called the BENEFITS-OF-PHILOPATRY hypothesis. And this hypothesis argued that it's not the cost or the constraints on independent breeding why individuals don't try to breed on their own. It's the benefits of remaining home, okay? And the idea was not based around means, it was based on variance. And so the whole hypothesis actually comes down to this idea that if you compare 3 species, an acorn woodpecker, which is a cooperative breeder from the American Southwest, the green wood hoopoe, which is a cooperative breeder from sub-Saharan Africa, and mountain chickadees, which are a non-cooperative breeder from this part of the world, and if you looked at the variation in reproductive success across territories, the coefficients of variation are much, much higher in the cooperative species than the non-cooperative species. Okay? Now, for many years, people argued about these hypotheses and then came to the conclusion that they're really saying the same thing. One is thinking about the benefits, and one is thinking about the costs. And if you think back to Hamilton's rule, we always have to think about the costs and the benefits. Okay, but one key difference that was overlooked for a long time is one hypothesis was based on the mean fitness, and the other hypothesis was based on the variance in fitness. And I think that's a critical thing to point out and think about. And so when we start to think about patterns of variance in fecundity or variance in anything, we think about bet hedging and bet hedging strategies. Okay? Now, we often think about diversified bet hedging strategies, or strategies that take advantage of alternative environmental scenarios. And this is quite simple. This is how individuals decide whether to literally put all their eggs in one nest, one basket, or spread them across different nests, across different oviposition sites. Okay? This is the way we approach our lives if you invest. Right? You should invest in a portfolio of stocks and funds rather than putting all your money into Google or Apple or Microsoft. You might want to spread it around 10 different things. Okay, this is diversified bet hedging, and this is what animals do. But there's another type of bet hedging that's much rarer. This is called conservative bet hedging. And these are conservative strategies that are thought to minimize fecundity variance across all possible environmental scenarios. What this means is you want to prevent complete failures. You want to cut off years of zero fecundity. Okay? Now, this idea is interesting but rare and somewhat contentious because it only applies to small populations. Okay? So when populations are small, the variance in fecundity can influence fitness as much as the mean. Okay? But most organisms don't have small populations because there's gene flow between groups, and they don't fit the criteria for this. But people have argued and shown theoretically that cooperative breeders may actually fit because you've got these subdivided populations with reduced gene flow. Okay, so there's these ideas that conservative bet hedging strategies can operate in these cooperative species. Okay, and what we find is if we look at the relationship between variance, here standardized, and breeding rainfall in this case, we find that among-year variation in pre-breeding rainfall does influence variance in reproductive success. Okay, so there's a negative relationship such that when conditions are good, the variance is a lot lower, and when the conditions are bad, it's a lot higher. So we have this idea then that social groups can have reduced reproductive variance across years. Okay, but the big question is still, does breeding cooperatively actually positively influence fitness? So what I mean is having helpers at the nest and living in the large groups confer these individual benefits. So this reproductive variance idea is on a group benefit idea. And to test it, we need many more years of data than I have on an individual level. Okay? So it's a hypothesis out there. Some people have tested it subsequently and have found support for it. But what we can do is go back to our long-term study and actually look at other measures of fitness that would be consistent with this idea. And this is work that my current PhD student, who's finishing on Monday, so will be a past PhD student, has been working on it for a number of years. And then she really wanted to come here for a postdoc and work with Ben, but she's continuing to work on the squirrel project, but based out of Canada, starting in about 3 months. So Sarah's been working a lot thinking about the cost. You saw the oxidative reproductive cost earlier, and also these fitness benefits. And I'm going to show you a few. So we've known for a while that having helpers is important. Okay? So this is comparing nests where offspring starve and where they don't starve and where they do starve. And those where there's starvation tend to have fewer helpers, and those helpers do less work. Okay? So having helpers basically increases the number of offspring that the parents will fledge. Okay? And having helpers also reduces predation on offspring and their fledgling success. Okay? So this is the number of helpers. It's just transformed. And if the number of helpers increases, the predation on offspring goes down and the fledgling success goes up. I've taken out the data just because it's really messy, but these are the significant trends that you see. Okay, so having helpers is important. There are direct benefits on fitness. Having more helpers is more important. It'll asymptote at some point, but we see basically more helpers is a good thing. And also, living in large groups is a good thing. Individuals in larger, above average group sizes tend to live much longer than those in small groups. Okay? So there are fitness benefits to rearing young. There are survival benefits to the adults. Basically, living in a large group with many helpers confers many fitness benefits, okay? So I've argued that environmental variation on very broad scales is correlated with cooperative breeding behavior across species. Breeding cooperatively positively influences your fitness, both your reproduction and your survival. But I want to go back to those comparative correlations that I showed you and ask another question, okay? Does environmental variation actually drive the evolution of social behavior, or does being social permit you to move into these harsh environments, okay? So we can't tell from just this correlational relationship. These data are consistent with that hypothesis, but I think it's a little bit more complicated than that. And so since we published all those comparative data, the methods have improved a little bit, and we can start to look at directionality in that. Okay? Now, why would I come up with this hypothesis? And let's leave birds for a moment and think about another group of social organisms, insects. Okay, so these are ants. And social insects in general, this is a number you hear thrown around a lot, make up half of the biomass of the planet's biodiversity. What that means is in the ecosystems where they occur, social insects often make up more than half of the biomass there. Okay, and this has been argued that these social insects are great colonizers and can take over environments. Not a lot of data to support that hypothesis. However, we've been approaching that with some other studies. So these are cooperatively breeding burying beetles. Okay? They're cooperative, not in the way that the birds are. They cooperate to bury a mouse carcass in which they lay their eggs. And the reason they need to cooperate is they're in rapid competition with flies who lay their eggs in the carcass. And if the flies get there first, the beetles basically won't reproduce. And so if they're cooperating, they can outcompete the flies. Okay? What this does is by cooperating, it allows them to expand their sort of ecological niche into the areas where the flies are most abundant and actually compete and actually reproduce. So by being cooperative, they can expand their range. They've also been thinking about this idea in the context of another species I study, eusocial snapping shrimp. I won't go into too much detail today, but these eusocial species, there's about 45 species in the group we study. About a third are eusocial. And the eusocial ones come to dominate the reef in terms of abundance, in terms of biomass, and in terms of number of host sponges occupied and the breadth of their host range. Okay, but it turns out that we've been looking at the relationships between their ecological niche, their how many host sponges they are able to use, and their transitions to social behavior. And we see the same sorts of things between these relationships between their niche and whether they can expand out and when cooperation evolves. So it leads us to this hypothesis where we wanted to look at the evolutionary transitions among these. In this case, we had about 4,500 species of birds between the environment and the social behavior. Which came first? Living in a harsh environment and then cooperation involved to take advantage of that area? Or did cooperative breeding evolve first and allow them to move into these harsher environments? Okay. What we find, so this graph is comparing non-cooperative and cooperative species and looking at their transition rates. You can ignore the two different dots. The paper is focusing also on monogamous versus promiscuous species and putting that into the story, but I won't go into that today. What we find is that transition rates from living in benign environments to living in harsh environments were twice as high in the cooperative breeders. Okay? So what that means is that cooperation evolved before the transition into these harsh environments. Okay? So living in a benign environment, cooperation evolved, and it seems to have enabled them to move into these harsh environments. Okay, so it supports that second hypothesis there, which is a little bit of a surprise and a little bit contradictory to everything I told you up until that point. So the story is a little bit more complicated. Okay? So environmental variation influences broad-scale patterns of social behavior. Cooperative species seem to occur in these harsher environments, but cooperative breeding also allows species to colonize the harsher environments. And then at a more sort of individual level, individual species level, cooperative breeding influences species differences in those oxidative costs of reproduction, and also the environmentally induced variation in reproductive success, and it also influences fitness and survival and reproduction. Okay? What I'm not telling you is that it influences survival and reproduction differently in good and bad years, and it's a complicated story. basically the pieces seem to suggest that having helpers is important in both bad years, but also in good years. Okay, so it's not just bad, but the survival in good and bad years, we're still trying to tease that apart. But the reproductive benefits are in both. Okay, so I gave you many examples of how social behavior might be related to environmental variation. But I want to step back. And behavior, I think, is just one way that animals can adapt to these unpredictable environments in which they live. And there are many other ways, and I suspect many of you in this room study some of those ways, okay? So I would argue that organisms use a variety of evolutionary responses to adapt to variable environments. Social behavior, cooperation is just one. And there are three general categories that we think of: plasticity, BET-HEDGING, which I introduced a little bit, and adaptive tracking, okay? So plasticity is probably an easy one for most of us to understand. This is the ability to respond to environmental cues and adjust your genotypic expression accordingly, okay? So some examples are things like Daphnia, when they grow in predator-rich environments, they get these helmets on top of their heads, and when they're in predator-free environments, they don't. This is really developmental plasticity, or what we're calling irreversible plasticity. It's laid down during development and it can't be changed. Another type of plasticity, though, is reversible plasticity, or things that change throughout the lifetime. There's many examples of this. I like this idea of seasonal coat coloration change in some mammals, okay? So they're dark in one time of the year and they're lighter in another time of the year when there's snow on the ground. BET-HEDGING, I introduced you to this idea, the idea of diversified strategies, and this diversified BET-HEDGING, when we think about how animals spread their eggs onto different oviposition sites or different nests. And I introduced the idea of conservative BET-HEDGING strategies, and maybe cooperative breeding behavior is consistent with one of those ideas. And then adaptive tracking, we can think of as a sort of a convoluted definition, as correlated variation in mean population traits as selection favors different mutations over time. What that means is that the traits are tracking the environment. I think the best example is our own evolution, okay? So we evolved in East Africa. Our ancestors came out of the forest at a time when there was climatic instability and variation, and over these long drying and wet periods, going back through time, we see shifts from dry to wet, and each time that there was a shift, evolutionary anthropologists have argued that there have been major adaptations, like coming out of the forest, bipedality, using fire, using tools, those sorts of things, being in groups, okay? So over long periods then, these things track these long-term environmental changes. So we asked a while ago, how can we put this literature together, right? Behavioral ecologists don't think enough about temporal variation. I would argue we think a lot more about spatial variation in the environment. But ecologists and evolutionary biologists think about this all the time, from oscillating selection in Darwin's finches to intermediate disturbance hypothesis, all sorts of things. We think about temporal changes, okay? But as behavioral ecologists, we need to think about how to put these together. And so we did this through a model where we modeled the joint evolution of a range of the evolutionary responses to environmental variation. I'm not going to go into huge detail in the model. It's published now. But it's an individual-based evolutionary simulation where we can basically give genes for each of these different types of responses, the plasticity, adaptive tracking, et cetera, and then model them using a reaction norm approach and allow mutation and natural selection to proceed. And so we can give different options. So organisms might have a fixed strategy. So we've modeled this in the case of temperature variation, but you could do it in terms of rainfall variation too. So there's a cue, something about the temperature in the environment, and insulation is here as the response, maybe the growth of their hair. So all individuals in a population will give potentially can give a fixed response. They could give a plastic response that's either reversible or irreversible, but as the cue changes, they can also vary the response. And then there are these mixed strategies or probabilistic, where some individuals in the population can do one thing, and other individuals in the population can do another in response to a given cue, okay? So this is getting at sort of a BET-HEDGING approach. And when you then model, you let your populations grow, and you model, or you vary a few parameters. The key parameters in our model are the time scale of environmental variation, so that's how an organism perceives the world. You perceive climatic variation very differently than a Drosophila, just because you live a lot longer. So a cycle for you is different than a cycle for Drosophila. okay? And then we can look at the predictability of that variation. And when you do that, and you model this, you get this really odd-looking graph that sort of looks like this. And basically, we can predict the parameter space, which is these two parameters, where you should make switches from reversible plasticity to irreversible plasticity to adaptive tracking to your types of BET-HEDGING. And you can use this model to do a whole bunch of things, thinking about tipping points with environmental change and whatnot. But I want to just illustrate now that for many organisms that are relatively long-lived and live in unpredictable environments, they're going to fall down here. And this is where we think the superb starling falls. Some are in this relatively long-lived organism, and we know, or I will show you some evidence in a few minutes, that there are different responses they can use, okay? And I want to illustrate that with one system, the vertebrate stress response, okay? So this is I'm going to simplify it, and it's a hormonal cascade from the brain all the way down to the adrenal cortex, where we actually get the release of the glucocorticoid corticosterone, okay? And these levels will circulate in the blood, and I'm going to talk about the receptors that they need to bind to, and a few other things. But most of what we know in birds about the HPA axis and the stress response comes from studies of species in the temperate or even the Arctic zone, right? I would argue that probably, 80 or 90% of the papers published have come from John Wingfield or his progeny over the last 40 years, okay? And most of that work came from thinking about white-crowned sparrows in temperate and Arctic regions, where we know that rapid and temporary responses to climatic perturbations lead to the production of these stress hormones, okay? In the tropics, things are very different. You don't often experience a snowstorm at the beginning of breeding, okay? You live in an environment that is very unpredictable every year, and you need to be cautious in how you do this, and maybe you need flexible adaptations to this continual climactic uncertainty. But we don't really know a lot about stress physiology in tropical birds, okay? We don't know how the stress response is even shaped by living in these variable and unpredictable environments. When it comes to the tropics and stress physiology, most of what we know comes from rainforests, okay? There's not a lot done in these semi-arid environments. And so I want to give you a little bit of data today to illustrate the hormone, the ligand, and then a little bit at the level of the receptor. And I'll illustrate using some examples from plasticity and BET-HEDGING. And I'm going to do it in the superb starling. So first I want to talk about the hormone, which we think is a reversibly plastic trait, right? They can change levels of hormones to change physiology and behavior throughout their lives. Okay, we know, going back to this long-term study, that CORT varies with climate, in this case among year variation in rainfall, but only in the subordinate individuals, okay? So in dry years, helpers have elevated levels of stress hormones, and in wet years, they don't. And the dominant individuals, the breeders and those non-breeder, non-helpers again, who are both dominant to the helpers, their levels of circulating baseline stress hormones or stress-induced hormones, which I'm not showing you, don't change from year to year, okay? It's only in the subordinates. This puzzled us for a long time, and we continue to work on this. I started this for my PhD in 2000, so it's been, 17 years. We still are working on the data to this, but all the data are consistent with the idea that the reason the subordinates have higher hormone levels is not directly related to the environment. It's because the dominants beat them up more in those years, okay? So in dry years, they just get picked on more, and the hormones are a response to the social environment. So social factors appear to influence CORT more than environmental ones within this single site over long term. But we want to approach this question from a different perspective. What if we looked at a bunch of different sites in a given year that differ in their climate, their long-term climate, their degree of predictability in rainfall? Okay, so we're moving away from analyses of time, one site looking across time, to analyses of space and looking at multiple sites in a given year and looking at their long-term and short-term climate. So what we've done over the last couple of years is set up this ecological gradient where we've got 9 populations across Kenya from the deserts in the north. This is massive desert. It gets about 150 millimeters of rainfall a year, all the way to some sites as you get out of the desert into the savannah, up to Mount Kenya where it's very wet, and all the way down to the Masai Mara where it's relatively wet, okay? So we've chosen sites that vary from about 150 to 1,500 millimeters of rainfall. The variation is different, and the predictability is very different. And we've just started this. I'm going to show you some preliminary data. We just got another year's data finished analyzing a week ago, and I haven't had a chance to look at it yet. But if you look at baseline stress hormones, those taken immediately upon capture, what we call stress-induced hormones when we measure the level 30 minutes after capture when it's at a peak, and then if we inject them with the hormone ACTH, which ramps up their HPA axis to the maximum. We relate these to different climate measures, and they relate to different timescales of environmental variation, okay? So baseline levels are highly correlated with annual rainfall, either what's fallen in the last 12 months, or you can bin it by 3 months, 6 months. So what's fallen in the short term predicts their baseline levels. But their stress-induced levels are highly correlated with the long-term mean at that site. Okay, so that's that range from 150 to 1,400 millimeters. And then the ACTH, when you inject them with this hormone to stimulate the HPA axis to its maximum, that seems to be most related to the long-term variation in rainfall. Okay? So different components of the stress response seem to respond to different timescales of environmental variation. Okay? So selection may be acting, different selective forces may be acting on different components of this HPA axis. And it's a plastic, each of these are plastic responses, but they seem to be shaped differently by natural selection. At least that's the working hypothesis. So now we're going back over different years, dry years, wet years, and repeatedly sampling these. And hopefully we'll have a more thorough answer soon. Okay, I want to switch gears and talk about what we think is an irreversibly plastic trait. And this is not with the ligand, with the hormone, but it's actually with the receptor. Okay, so superb. Up until this point, we focused primarily on events that happen in the adult lifetime of these birds, okay? But we can also ask what happens to birds when they're born in a dry versus a wet year. Do these have long-lasting consequences, okay? Do early life conditions influence physiology and fitness? We come at these questions both from an adaptive perspective, but also from a mechanistic perspective. We know from studies of mice and rats that early life conditions can influence behavior and physiology. And one way that this is done is through the addition of chemical modifications to the DNA called DNA methylation. Okay? So when rats are licked and groomed more as babies compared to those that are not, they have differences in levels of DNA methylation in parts of their genome. One of the key regions is the glucocorticoid receptor. So this is the receptor where that hormone corticosterone binds to. Okay? We can ask for animals living in unpredictable environments where early life conditions are not predictive of what they're going to experience later in life. How do those environmental stressors during early life influence their fitness? And what are the mechanisms? Could this DNA methylation be underlying some of this? There's really 3 hypotheses that you can put forward. The first is that called the predictive adaptive response hypothesis, where individuals can adjust their phenotype throughout life to match the current conditions. There's a developmental constraint hypothesis, which basically argues that the conditions you're born under constrain your fitness later in life. And there's a developmental programming hypothesis, which suggests that individuals maybe born in those low quality, poor conditions might be able to give an advantage by their mother through some sort of maternal effect that would allow them to improve their fitness later in life. So for species that are born in these unpredictable environments, the first hypothesis doesn't really apply. So I'm going to focus on these last 2. Okay? And what do we think is happening? Well, we know that DNA methylation, how this can work is that when the upstream regulatory region of a given gene is unmethylated and normal, it will help the gene be expressed. But if you put these methyl marks on these certain sites in this regulatory region, it can block gene expression. Okay? So there's a relationship potentially between these methyl marks and expression of the downstream genes. And we can start to make predictions. So from the developmental constraints hypothesis, if individuals are born in poor, low rainfall years, they're likely to have lower fitness later in life. And that might be because of increased epigenetic regulation. In contrast, the developmental programming hypothesis suggests if individuals are born in poor years, mothers might be able to do something to give them an adaptive advantage later in life. And we might see a downregulation in the expression of DNA methylation. Well, what did we do? This was the first attempt to do this in birds. It took a long time. And as I told some people today, I'm not sure I would recommend anybody trying this approach in the future because it's problematic in some ways. But we basically sequenced the upstream region of the superb starling genome, and we found a region that was homologous to the promoter in rats, and it's partially overlapping with what we think is the promoter in starlings. Okay, and then we sequenced this. You can see each of these blue areas is a CpG, a C next to a G, where you can get these methyl marks. So there's a lot of them in here. The blue is the putative starling promoter, the yellow is the homologous rat promoter, and the green is where they overlap. And then we look at methylation levels, okay, across not only the upstream regulatory region, but also this is the first exon. Okay, methylation is much higher in the exon, which is to be expected, and much lower in the upstream region. We're doing this from blood. We're not doing this from brains because, A, we can't kill birds in Kenya, and B, we don't want to kill our long-term study birds. We're looking at them when they're born, and we want to follow them throughout their lives. And if we zoom in on the area where we think the promoter is, there is some variation, and it's low overall. The methylation in each of these CPG sites in this regulatory region is correlated with the sites next to it to some degree. So I hesitate to call them differentially methylated regions, but they're sort of analogous to those. So this is in the homologous rat promoter, and if we look in that starling area, we see 3 areas of correlated CPG sites. Today I'm just going to talk about the most upstream region. I'm calling region A. If we look at that region, we can find a few things. So this is comparing DNA methylation in chicks. We always sample chicks on day 7, and then we often sample birds later in life. A few weeks later, up to 12 years later. They live probably 10 to 15 years in the wild. And these methylations, so this is the difference between their initial capture and their second, and there's really no change over their lifetime. Okay, so methylation does not seem to be changing, so we think it's an irreversibly plastic trait. And what's interesting is that the amount of methylation is positively related to pre-breeding rainfall. Okay, so birds born in dry years or following dry years tend to have low methylation, and birds born following wetter years tend to have higher methylation. It's subtle, but it's a significant relationship. And we also find that birds born in dry years are actually more likely to breed than those born in wet years. And this was puzzling to us at first. Okay, so you're born in a dry year, you're more likely to breed over the course of your lifetime. And whoops, and there's a slide missing, but there's also then the relationship between the methylation and the likelihood of breeding in your lifetime. Okay, so what we find is that when environmental conditions are poor, individuals tend to have lower methylation and a higher likelihood of breeding over the course of their lifetime. Okay, so it's very preliminary. It's a first step. There's a lot we don't know. We don't know what role mothers play in all of this. Birds are nice because they provision the eggs with various things, and so we're testing these hypotheses now about what mothers may put in the eggs that could influence DNA methylation, these maternal effects. And we also don't know the relationship between DNA methylation and rest of the HPA axis, both the expression of the receptors, but also the role that corticosterone plays. And we're starting to get at this by working locally on European starlings, where we can actually take their brains and do these proper comparisons. But what we do know is that there seems to be at least a weak relationship between the amount of methylation in these birds and their baseline stress hormones. Okay, so birds that have lower methylation on day 7 tend to have higher circulating corticosterone during those times too. Okay, I'm going to give you one final example, and it's an example of a BET-HEDGING response, and again looking at the receptor. So we can ask, does this glucocorticoid receptor, which is highly conserved across vertebrates, show signatures of natural selection? If so, do they relate to patterns of environmental variation? And this is work that a former master's student, Natalie Hofmeister, who's now a PhD student at Cornell, did for her master's work. Okay, so we went back to this group of African starlings. Remember, there's 45 species. We were able to get good DNA from about 30 of the species. We were able to sequence all of the exons of the glucocorticoid receptor. So we're working on the genes here, not the regulatory region. Okay, and what we find, actually we sequenced 7 of the 8. We couldn't sequence this one, exon 3. We find basically very little evidence of positive selection. Okay, so that would be DNDS ratios greater than 1. Most of the DNDS ratios are less than 1, and there's only 2 codons in this N-terminal region that seem to be under positive selection. Okay, in the 30 or so species we looked at. Okay, so there's very little. There is variation, but it's not a variation that seems to be under positive selection. Okay, so we can start to make some predictions, though. Environmental variation and DNDS ratios. If we see a positive relationship, the environmental variation is basically calculated as this bird's range, calculating the average, mean, and variation of rainfall and temperature variation across its range, and we can relate that to the DNDS ratios. If we see a positive relationship, it would be suggestive of an adaptive tracking response, and if we see a negative relationship, it would be suggestive of genetic canalization, which is analogous to a BET-HEDGING response. And to our surprise, and I say this is a big surprise, we see a very strong relationship. There's a negative relationship between the variance in precipitation across a bird's range and its DNDS ratios, okay? Which is consistent with this idea of genetic canalization. This kind of makes sense. right? This receptor is highly conserved across animals, and it's highly conserved within starlings. And it's probably the variation in the ligand, the hormone that binds to it, that's more plastic. Okay, so I've argued that we can look at the HPA axis, and different components of it might be thought of as different types of adaptive responses, and they're under different types of selection. Okay, so hormones are reversibly plastic, but different components, baseline, stress-induced, et cetera, respond to different timescales of environmental variation. The methylation at the receptor seems to be irreversibly plastic and influenced heavily by early life conditions. And then genetic variation in that receptor is low and seems to be canalized, but it's related to patterns of environmental variation, okay? So when we think about these environments then, where this is the picture taken the day before that rained and turned green. It was really brown, and as I mentioned, it turned green the next day. So these types of environments occur across the globe. Much of sub-Saharan Africa and these semi-arid environments, some people would argue 20 to 25% of the globe experiences these types of environments. So organisms have evolved in these unpredictable environments for millions of years. And I think by studying them, we can get a better understanding of the functional significance of behavioral and physiological adaptations that allow them to respond, and then we can start to make better predictions about how organisms everywhere are going to respond to climate change. So with that, I'd like to thank a bunch of collaborators, NSF, and for you for listening and inviting. Thanks. Yes?
Yes, thank you for an interesting talk. Let me ask you a question about the model that you had. Can you go back on the?
Yeah.
Can you go back to where you have the parameter space study there?
Mm-hmm.
Yeah. So I couldn't quickly look at the whole thing, but I wonder if you thought about applying Levins's theory of adaptive functions, where you have a fitness adaptive function associated with it. Because it appears to me, looking at that just briefly, the adaptive tracking would correspond to a convex fitness set with a linear adaptive function, and the other two, the blue and the red, would correspond to concave adaptive functions with either linear or hyperbolic adaptive functions. I'm not sure about the other two sections. Have you thought about maybe taking that kind of an approach?
We've thought about, we've been thinking a lot about what the next steps of this should be. Our first approach was to test this because it makes some simple predictions. But yeah, our relationships with fitness in this model, which I didn't show you, are relatively simplistic, and I think you could vary those relationships quite easily and get different trends.
Sort of a more analytical way of looking
Yeah,
at it.
yeah, it's a good idea. We haven't done it yet. Yes.
You've explored, it looks like, genetics and physiology of these adaptive responses of these long-lived animals. Have you studied learning at all?
I have not.
Or is that part of your research program? Or do you think that would be a useful thing to add to this? Starlings are part of a group of birds that are fairly smart. And,
Yeah. No, I haven't studied learning.
and I just wonder how that might contribute to this whole.
Yeah, I've had a bunch of students that have thought about doing that. One of the reasons we haven't done it is because they're too smart. They're really hard to recapture. They learn really quickly how to avoid nets and traps, and so to handle them, to do some experiments has been tricky. To do that, I think we would want to get more experimental. We haven't done it. I think there's evidence from other cooperative breeders, in particular, that they're good learners. I mean, we're starting to get into sort of social brain and social cognition hypotheses, and there's very mixed literature on the relationship between that and complex social behavior. But it's also a good idea. Oh, yeah.
I have a question about the correlation between the environment variation and the adaptation. It looks too good to be true. I wonder if there are other studies showing similar results, and how do you control the genetic distance when you calculate the DNS?
Yes. So it's done. The analysis is done phylogenetically controlled. Okay, so it's doing a phylogenetically controlled least squares regression. So it's controlling for genetic differences between the species. And yeah, it looks it looks too good to be true. I was really surprised, especially because we have no evidence of positive selection. I thought we might see evidence of positive selection and it may be related to. But you know, we looked at a bunch of different environmental parameters and they don't predict it. This one does predict. So our models can include, you know, a bunch of other things. I'm just showing you the one that was most significant. And it's all done in a phylogenetically controlled context. So we control for genetic distance between species. Ben?
Yeah, so I'm just wondering about the methylation differences for low treatment, pre-treated rainfall, chicks born in the lower methylation, higher probability in the future or in that year?
In the future, in their lives.
But like, how confident are you in that? It's just some
Not very confident.
visible misinformation.
Right. So I mean, that was a sort of a first pass. We're trying to look at some other genes and players in the HPA axis, and then we're also taking a whole genome approach where we're looking. We're comparing birds born in wet and dry years and looking at global methylation across the genome. Okay, so we've done that. I was hoping to have that. And so we've compared. Right now we've compared 12 individuals born in wet and dry years as sort of a pilot study, and we get very good coverage across the genome. We pick up the GR receptor, a bunch of other things, because we've selected for CPG islands, which are likely to be these promoter regions. And the bioinformatics is complicated, and depending on which programs we use, we get sort of different results. I mean, we're getting consistently, I think at this point the one we trust the most is we're getting about 30 or 40 differentially methylated regions. We're still probing what the genes are in those regions, but what's popping out most consistently are things related to cardiac function. I'm not sure why. There's like 3 or 4 genes in different chromosomes, different parts of the genome that seem to be differentially expressed. At that level, GR is not popping up, but that's because we have a small sample size. Could be because we have a small sample size and we don't have a lot of power to detect it. So, you know, it's different approaches. When you use a targeted gene approach, in some ways you have a little bit more power. When you use a whole genome approach, you don't have a lot of power. We're trying to use both, and hopefully the results will be consistent, but we're still a couple months away from figuring that out. So, yeah, I said I mean, we got a significant result there. You can take it for what it's worth. It's not, it took many years, and it's not necessarily the best approach, I think, to take a targeted gene that we know plays a role in many things in this species and look at the methylation. It's hard to get the data, and B, I think the whole genome approach is going to be better.
Okay, last question.
So, expecting that the major way most birds deal with increased reproductive success and oxygen migrating. So, either in your starling context or your global studies, have you thought about trade-offs between different kinds of movement behavior and function?
Yeah, we didn't do it because the data we used for those comparative studies was compiled by Andrew Coburn. He looked exactly that question, and cooperative breeders tend to be non-migratory and more sedentary, and so they don't tend to move. What was that?
I was also curious, where is that?
Oh, as here as a,
Yeah,
where would something?
tracking or sort of leaving a place, coming back,
I don't know where that one would fall there. I'd have to think about that one. But I think, you know, I think the interesting question is why don't cooperative breeders move, and why are there not a lot of nomadic cooperative breeders? Okay, there's a lot of nomadic birds in sub-Saharan Africa, and they move around and they follow the rains or they come back. The non-cooperative species that we study, we don't study them as well. They kind of disappear during the non-breeding season. Like the superb starlings will come back to their site at least once a day. I say that, except this season that they're in a 50-year drought, and I have a student who's been trying to work in the dry season, and there were no birds on half of the sites. We don't know where they went. They literally disappeared. Usually we can talk to people and find them. So even these cooperative ones have a sort of partially abandoned their site during the dry year. So there's some flexibility there, but why they don't just move, I don't know. I mean, I think that the hypothesis that I would have is that the territories, I talked a lot about temporal variation. Spatial variation is really important. The territories that they can occupy and breed successfully in are quite rare. And so they will basically have these dynasties on these territories probably for hundreds of years. And I think if they migrated and came back, someone else might be there and they have to fight for them. So they occupy these things year-round because they need these territories to survive, to have enough food during the breeding season. And I think if they moved, either nomadic or they migrated, then they wouldn't necessarily get those. And I think I alluded to that with the group size. There's the survival benefits of being in large groups. There's also probably competitive benefits in being in large groups. You can't be displaced, whereas in some other plural breeders with slightly smaller groups, there's a lot of more fights between groups and they displace them. So I think the selection on movement would probably be because you lose your territory and then you lose your, and to answer that question, and people have looked at this in other species, you need to think about genetic lineages through time, okay? So these share, you know, if you're on this territory and producing offspring for 100 or 200 years, you could think about it in those terms of genes being passed down.